So what I'd like to talk to you about now is I'd like to focus a little more on what's happening at these replication bubbles. So let's go ahead and take a close look. So we have a piece of DNA here. And remember, to have replication occur, we have to have an origin of replication. It's really important that we have this origin present. So let's say I'll label my ends here. Here's the 5' end, here's the 3' end, which over here, of course, means I've got 3' and 5'. So there's my piece of DNA. Here's my origin of replication, and I'm now ready to start replicating this DNA. So as we start replicating, what we're going to see happen is this origin is going to open up and replication is going to proceed in both directions. Okay? And we call this structure a replication bubble. Now every replication bubble has two replication forks. One here and one here. So here is one replication fork and here's the other replication fork. Okay. So let's go ahead and take a closer look at one of these structures. So we're going to zoom in on this replication fork right here. So label my 5' and 3' ends. There we go. Okay, so here's my replication fork blown up in size so we can take a closer look at it. Now there are going to be several enzymes and proteins involved in this process that are ultimately going to facilitate replication. These all come in to essentially help open up this fork so that we can then start replicating the genetic material. So the first enzyme that's going to be utilized is an enzyme that is going to unwind this double helix of DNA. So we still have unwound DNA here, and this enzyme that's going to be responsible for unwinding the helix is called helicase. So there's helicase. It's going to move in, and it works kind of like a zipper, unzipping this double helix. Now, a couple of other things happen as we unzip this helix. First and foremost, we have these two strands that originally were associated with one another through hydrogen bonds. The problem right now, as it exists, is if this strand comes in too close a contact with this other strand, they could reform those hydrogen bonds. And we don't want that because that's going to slow down our ability to duplicate each of these strands. So there are going to be special proteins that are going to bind to each single strand, and they're going to prevent these strands from reassociating with each other. And we call these single strand binding proteins, or SSBPs. So these single strand binding proteins are going to be used to stabilize these single strands and keep them from coming back together. All right? Now, a couple of other things we need to consider. One of the big things we need to kind of keep in mind here is that as we're unwinding the helix with helicase, this portion of the double helix is actually becoming a little more tightly wound. And that's a problem because if it becomes too tightly wound, helicase is not going to be able to unzip it further. So there's a class of molecules called gyrases that are responsible for binding to this DNA here and cutting it and rearranging it so that it doesn't overwind. And the one that is used in our book is more specifically called Topo Isomerase. So the purpose of Topo Isomerase is to cut the DNA, reorient it, and prevent it from overwinding. So what have we done so far? We have started to unwind our double helix using helicase. We have bound to each strand using these single strand binding proteins, and we've now bound Topo Isomerase and cut this molecule at the region that hasn't been unwound to prevent overwinding. There's one final thing we're going to do before we're ready to start synthesizing DNA. And that last thing that we're going to do is we're going to bring in an enzyme that is going to be responsible for laying down small segments of RNA. These small segments of RNA are commonly called primers. And RNA, again, is going to run anti-parallel to our DNA strand. And the enzyme that's going to be responsible for laying down these primers is called primase. This enzyme lays down these primers because without these primers, DNA polymerase, the actual enzyme that would synthesize the DNA, will be incapable of binding and synthesizing DNA. You can kind of think of these primers as starting lines. This is essentially telling the polymerase where it needs to begin replication. So polymerase will bind here to the 3' end of this primer, and it will synthesize in this direction. And another one will bind here and synthesize in this direction. So these are the steps of preparation that we have to go through before we can even bring polymerase in so that it can start synthesizing the genetic material. Okay? So let's go ahead now and take a closer look at what's going to happen on each of these strands as the polymerase begins to elongate the DNA. All right, so I'm going to go ahead and erase this stuff. All right, so let's go ahead and redraw our fork. I'm going to redraw those primers. So there are my primers. And again, just to label those ends of those primers, here's the 5' end and here is the 3' end. 5' end and 3' end. Okay, so DNA polymerase, which is the enzyme responsible for synthesizing new DNA, in this case specifically DNA polymerase 3, needs a 3' end to attach to. So it has to be able to find a 3' end on a new piece of a nucleotide or polynucleotide. And so this 3' end of my primer serves that purpose. So polymerase is going to bind here. So this is polymerase 3. And polymerase 3 will also bind here. Okay, so polymerase 3 binds here and here. This polymerase 3 is going to begin moving in this direction because polymerases always synthesize the new DNA from the 5' end towards its 3' end. So they're always adding to the 3' end of the molecule. So as it synthesizes DNA, it's going to be moving in this direction. This polymerase up here, though, is going to be synthesizing new DNA in this direction. Okay, so you'll notice each one is moving towards a 5' end on its template strand of DNA, but it's synthesizing the new DNA from the 5' end to the 3' end. Now, what you may also notice is that this particular polymerase is moving into the replication fork, while this polymerase is moving out of the replication fork or away from it. And so we have names for these two strands. This strand on top is commonly called the leading strand, and the strand on bottom is called the lagging strand. Now, the reason this is called the lagging strand is because as the polymerase is synthesizing this DNA, the helicase is still unwinding the replication fork. And so what's going to wind up happening is we're going to wind up opening that replication fork even more. So we're going to open it up a little more. And as a result, this primer is actually right about here. And so this polymerase has now synthesized new DNA up to this point. But while it was doing that, the fork opened a little more, and another primase jumps in and lays down another primer. This polymerase on the top, however, because it's synthesizing into the fork, it can just continuously make DNA as the fork opens up. And so what you wind up getting is you wind up getting these two different scenarios. In one, the polymerase just continuously moves along the top strand, or whichever strand is the leading strand. It's not always the top, but in this case it is. So it moves along the leading strand, synthesizing our new molecule of DNA, moving into the replication fork. On the other hand, you have a polymerase that's going to synthesize in this direction, and then once it reaches the end here, it's going to hop back, bind here, and synthesize another fragment of DNA up to the previous primer. So this is why one strand is called the leading strand and the other is called the lagging strand. The leading strand is one where the polymerase can continuously move into the replication fork, and the lagging strand is one where the polymerase is moving away from the replication fork, and as a result, as the fork opens more and more and more, we lay down primers close to where the fork is opening up, and the polymerase consistently jumps backwards to synthesize yet another fragment. And again, we have a name for these fragments. They're called Okazaki fragments. Now, after we're done synthesizing all of this material, so after we have fully opened up and synthesized portions of this, there's going to be another event that happens. Namely, there's a gap here between this DNA and this RNA primer, but we also want to replace that RNA with DNA. We don't need RNA in our DNA sequence. We want it all to be DNA. So, what's going to happen is there is yet another polymerase enzyme called polymerase or DNA polymerase 1. DNA polymerase 1 is going to come in anywhere we have RNA sandwiched between two pieces of DNA, and it's going to replace that RNA with DNA. But there's still one minor problem. There's a gap that exists between our two pieces of DNA. So DNA polymerase 1 swings in, but we still wind up having a gap, a break in our new strand of DNA. We want to link these Okazaki fragments into one consistent piece like we have on our leading strand. So after DNA polymerase 1 swoops in and replaces the RNA with DNA, there is another enzyme that's going to come in, and it's going to link these fragments of DNA together. And this particular enzyme is called DNA ligase. It's called DNA ligase. Okay? So DNA ligase is the enzyme that swoops in, and it's ultimately going to link together those fragments of DNA. So polymerase 3 lays down the initial pieces of new DNA that we synthesize on both the leading and lagging strand. DNA polymerase 1 is going to swoop in, and anywhere there is an RNA primer sandwiched between two pieces of DNA, it's going to replace that RNA with DNA, and then ligase is going to come in and it's going to link the fragments, those Okazaki fragments, together, giving us one long consistent piece of DNA with no breaks in it. Okay? So this is what's happening at the replication fork in DNA replication.